Okay, good morning, everyone. Welcome to the third day of the HydroMediT 2024 conference. This is the first oceanography session for the day. And without further ado, I say, let's start with the presentations. The first one is by Ms. Stivachti, Yannoudi, Krasakopoulou. And the title is Investigation of the Response of the Saronic Gulf CO2 carbonate system to changes in dissolved inorganic carbon and total alkalinity. Mr. Vakti, the floor is yours.
Good morning, everyone. Thank you for being here, Saturday morning. So today I'm going to present to you our study on the investigation of the response of the Saronic Gulf carbon dioxide carbonate system to changes in dissolved inorganic carbon and total alkalinity. As we all know, the ocean has the ability to absorb atmospheric carbon dioxide. As the world's largest carbon reservoir, it mitigates climate change through the uptake and storage of CO2. The excess CO2 into the ocean causes changes in marine chemistry. The parameters of marine CO2 system are total alkalinity, dissolved inorganic carbon, seawater's pH, and partial pressure of CO2. In order to investigate the marine CO2 system, at least 2 of the 4 parameters are needed. Through a series of chemical reactions, the ocean CO2 uptake leads to a domino effect that is an increase in CO2, a reduction in seawater's pH, and then to a reduction in carbonate ion concentrations, which expresses its main buffering capacity. DIC and bicarbonate ions increase as well. These changes in marine chemistry, referred to as ocean acidification, have a serious impact on marine ecosystems and its organisms, and on the efficiency of the ocean's CO2 uptake as its buffering capacity will be reduced. Marine organisms that produce calcium carbonate exoskeletons are more vulnerable to ocean acidification. This calcification ability can be assessed through the estimation of the saturation state of carbonate minerals. Values of omega for aragonite and calcite, which are higher than 1, indicate supersaturation, namely favorable conditions for calcification. And values of omega, which are lower than 1, indicate undersaturation. In order to quantify the sensitivity, the ocean's ability to resist changes in DIC and total alkalinity, a complete set of buffer factors was proposed by Eggleston et al. The 6 buffer factors quantify the sensitivity of aqueous CO2, hydrogen ions, and carbonate saturation state to changes in DIC or total alkalinity when the other parameter is kept constant. Specifically, buffer factor gamma expresses the fractional change in CO2, whereas beta expresses the fractional change in protons, and lastly, omega expresses the fractional change in carbonate ions. The subindex I expresses the change in parameter when the other remains constant. This process occurs during the air to sea CO2 exchange or additions of strong acid or base. In addition, Revel factor is also used to assess ocean sensitivity to an increase in atmospheric CO2. Minimum absolute values of buffer factors denote lower buffering capacity, while lower Revel denotes higher buffering capacity. The buffer factors have dimensions of micromole per kilogram, while Revel factor is dimensionless. The Mediterranean Sea is an important anthropogenic carbon storage area. From the 1st of May 2025 will be an emission control area, and the strict regulation of 0.1% of sulfur content will be established. Because of the price difference between heavy oil and low sulfur fuels, quite a few marine companies are expected to install exhaust gas cleaning systems, that is scrubbers. As a result of scrubbing process, it is predicted that large volumes of acidic water will end up into the marine environment, causing a reduction in total alkalinity. To this end, data collected in 2021 in Saronikos Gulf, which hosts the Piraeus port, one of the largest ports in the Mediterranean Sea, and the response of CO2 carbonate system to changes in DIC or total alkalinity was assessed. The current buffering capacity was quantified and compared with corresponding values of the Aegean Sea, and the future change in buffering capacity was assessed following a quite simplistic approach. About the methods followed, seawater samples were collected from 7 stations in the Saronikos Gulf. The sampling was carried out at depths of 2, 10, 20, and 50 meters. Seawater's carbonate system parameters, the Revel factor, omega-4 aragonite and calcite were derived from DIC and total alkalinity using CO2C's Excel program. The 6 buffer factors were calculated using the equations from Eggleston et al. As they modified by Alvarez et al. And the buffering indicators that affected by the reduction in total alkalinity were recalculated for the cases of 2 and 10 micromole reduction in total alkalinity. Those 2 cases of TA reduction were collected from the results of different simulations of Turner et al. That showed a reduction in total alkalinity in the Baltic Sea due to the extensive use of scrubbers. We chose those 2 cases despite the differences in biological characteristics between the Baltic Sea and the Mediterranean Sea, and assumed that DIC remains constant at 2021 values. As we can see, the results show that the mean concentrations of DIC and especially total alkalinity are higher than the relevant values of the upper layer of the Aegean Sea. Omega-4 aragonite and calcite are well above 1 and quite comparable to the Aegean Sea values. The absolute values of the 6 buffer factors are overall higher than the corresponding values of the Aegean Sea, while the Revel factor is lower. These results indicate higher buffering capacity of the Saronikos Gulf and less sensitivity to changes in DIC and total alkalinity. In the case of a 2 micromole reduction in total alkalinity, DIC over TA ratio and the Revel factor increased, implying a slight decrease in the CO2 buffering capacity. The buffer factor beta TA decreased, showed, sorry, excuse me. The buffer factor beta TA and omega TA decreased, showed the highest reduction, and the saturation state for aragonite decreased twice as much as the saturation state for calcite. In the case of 10 micromole reduction, which is an extreme scenario for the Mediterranean waters, the buffering indicators showed a higher reduction in the CO2 absorbing capacity. The buffer factor beta TA and omega TA decreased equally and more than gamma TA, and both of the aragonite and calcite saturation state decreased equally. However, their values remain far higher than 1. These results indicate more sensitivity to changes in pH and calcium carbonate saturation state than in CO2 for a given reduction in total alkalinity. In conclusion, firstly, about the current buffering capacity, the values of the different buffering indicators showed that the Saronikos Gulf's waters are resistant to changes in DIC and total alkalinity. The way that the absolute values of the 6 buffer factors are arranged indicates that the sensitivity of CO2 is the highest, followed by the pH change, and as the omega DIC and omega TA are the highest absolute values, confirms that Saronikos Gulf, like the entire Mediterranean Sea, is able to regulate changes in calcium carbonate saturation state. Secondly, about the future buffering capacity, and despite the simplistic approach taken to assess the future change, the acidification of seawater by strong acid causes a reduction in carbonate ion concentrations, which expresses its main buffering capacity. This is a process capable of turning the Saronikos Gulf into a source of CO2 in the atmosphere. Thank you.
And thank you for keeping the time. So let's proceed with questions from the audience. Do we have a question from the audience? Okay. Yeah.
For the very nice talk and this very important work for all the biogeochemical people. And I wanted to ask you, is this a normal situation? I mean, your final finding is that it potentially it will become a source. What about other areas of the Greek, let's say, seas? Probably it's, I mean, this is something common that we expect for the Greek seas, or it's the fact that it's it's an enclosed gulf and yeah.
Happened and the other.
Yeah.
But we investigate here the scrubbers.
Yeah, okay.
Any change of total alkalinity will cause this result.
Yeah.
We have this result.
Because of the reduction in total alkalinity.
At the reduction of total alkalinity.
But we have seen here that the reduction of these buffer factors was less than 3%, even at the worst scenario of alkalinity change of 10 micromole per liter. And this shows that Mediterranean waters and Saronikos Gulf waters have a high capacity to buffer this change.
Buffering capacity.
Yeah.
They will change the characteristics of the carbonate system, but
not
at a high rate as it will happen or it happens at the Baltic.
Okay, this is what
and this is due to the special characteristics of the Mediterranean waters that are originally
high total alkalinity,
characterized
warm waters and high total alkalinity.
Yeah. Thank you. This is a very, very nice.
Do we have any other questions? Okay, can I shoot one? Can I ask something? Okay, this is a kind of a box model approach, right? No?
No.
Okay, so that renders my question irrelevant then. Okay, I wanted to say if it's appropriate to approach the subject in a two-dimensional way, spatially. It is capable. You're capable of doing it. Okay. Are there any?
No problem. It's all in the Excel.
Okay, the routing, let's say, you can run it in a two-dimensional way, covering areas. Okay. So, thank you for a very nice presentation.
Thank you.
Let's move to the next one. Going to be delivered by Miss Skilaki. Okay. Co-authors are Parinos, Hadjianestis, Kambouri, Stavrakaki, Velaoras, Michalopoulos, and Gogou. The title is Downward Fluxes and Sources of Polycyclic Aromatic Hydrocarbons in Settling Particles from the Aegean in the Ionian Seas, Northeastern Mediterranean.
Okay. Okay, so good morning, everyone. My name is Ichthyology, and I am a PhD candidate at the University of Crete. Today, I will present your study on the downward fluxes and sources of polycyclic aromatic hydrocarbons, or else PAHs, in settling particles from the Aegean and Ionian Seas in the northeastern Mediterranean. So what are PAHs? PAHs are persistent organic pollutants found widely in the environment. Known for their toxic, mutagenic, and carcinogenic properties, 16 pH compounds are classified as priority pollutants by international environmental agencies. These compounds originate mainly from three sources: pyrolytic processes, like burning biomass and fossil fuels, petrogenic sources, like unburned petroleum, and diagenetic processes. Now, once hydrocarbons enter the marine environment, they undergo various alterations. Processes like photooxidation, dissolution, adsorption-desorption in particulate matter and marine sediments, but also biodegradation by marine organisms, play crucial roles in their transportation and in their transformation and transport to the deep ocean layers. So what is our main goal? This study aims to identify the major pH sources, to determine their downward export rates to the deep layers of the three major basins of the northeastern Mediterranean Sea, and to examine the biogeochemical and physical dynamics in shaping their composition, abundance, and spatiotemporal distributions. So in order to achieve that, we collected in the Greek National Project Gripis for integrated observatories in the Greek seas, we collected 3 sediment trap samples from 3 different sites in the northeastern Mediterranean between January to December of 2015. Now, the first site is in Athos Basin in the North Aegean Sea at 500 and 1,000 meter depth. The second site is in the Cretan Sea in the South Aegean at 350 and 1,550 meters depth, and then in the Ionian Sea at 700 and at 4,300 meter depth. Now, our study area, the northeastern Mediterranean Sea, and especially its coastal area, is a region under intense anthropogenic pressure, resulting in pollutant discharges. Now, the open northeastern Mediterranean Sea, being an important region for merchant shipping and oil transportation, it receives substantial amounts of petroleum discharges, while atmospheric deposition is another important pathway for the introduction of hydrocarbons, including significant inputs from Saharan dust, especially during dust storm events, that contains both natural and anthropogenic hydrocarbons. Now, in the map above illustrates the main water mass circulation pathways in the northeastern Mediterranean Sea, along with the classification of atmospheric deposition sectors. The map below annotates the possible oil spills detected in the Hellenic seas, along with the main tanker crude oil shipping routes. Now, in terms of its biogeochemical features, the northeastern Mediterranean Sea is characterized as an oligotrophic region, where the oligotrophy increases from north to south. Now, regarding the analysis, organic compounds were extracted from the freeze-dried filters and then fractionated into individual column classes by column chromatography on silica gel. The instrumental analysis was carried out by GCMS, and then we quantified 25 pH compounds, including 15 parent PAHs, along with their alkylated homologs. Now, moving on to the results, let's take a closer look at this graph, displaying a typical molecular profile of PAHs within the settling particulate matter. This graph showcases the percentage distribution of PAHs based on their molecular weight, offering valuable insights into their composition. Here, we can observe a mixed contribution from both higher and lower molecular weight PAHs, indicating a mixed contribution from different sources. So more specifically, when examining the lower molecular weight PAHs, such as those within the phenol-3 series, we observed a notable presence of substituted derivatives. This observation hints towards the potential petrogenic origin for these compounds, while the high prevalence of higher molecular weight PAHs suggests pyrolytic sources.
Now, concerning the molecular profile of the examined PAHs across the 3 sites, phenanthrene and its alkylated homologs dominated amongst the low molecular weight PAHs, peaking at the parent phenanthrene, while an exception is observed in the North Aegean Sea, where a peak occurs at the alkylated homolog rather than the parent compound. Now, among the high molecular weight PAHs, benzofluoranthenes, benzoperilene, and to a lesser degree indinopyrene were dominant in all cases. Now, regarding the temporal and seasonal variation on PAH fluxes, significant fluctuations were observed across the 3 investigated areas. The North Aegean Sea exhibited fluxes 4 to 5 times higher than those in the Cretan and Ionian Seas, as indicated by the secondary axis for this region, and higher fluxes were observed during late winter, early spring, probably due to increased productivity in the upper water column, organic matter export linked to winter convective mixing, and phytoplankton blooms. Then we used PAH diagnostic ratios to assess specific sources of PAHs across the 3 investigated areas. Low molecular weight PAHs, such as the phenanthrenes ratio, pointed out to unburned fossil fuels and more specifically mixed contribution from petroleum and combustion sources. High molecular weight PAHs pointed out to a mixed contribution from petroleum, grass, wood, and coal. Now, given the differences among the 3 study subregions, we analyzed each one separately. So, the North Aegean Sea, as I already said, exhibited the highest fluxes among the 3 sites, with petrogenic sources being dominant from unburned fossil fuels, probably due to heavy merchant shipping, oil transportation, and inflows from the Black Sea. Now, the mesotrophic character of this area probably enhances the petrogenic signal observed even in deep waters, since low molecular weight PAHs, that they are more abundant in the dissolved phase, are more efficiently scavenged by organic rich particles such as phytoplankton and fecal pellets, preserving them as they sink. Now, in the Cretan Sea, 3 peaks observed in the average pH fluxes in spring and fall. Their composition indicated a mixed contribution of both pyrolytic and petrogenic sources, with pyrolytic origins being dominant. This aligns with findings that links PAHs from fossil fuels and combustion processes to fine particles like char and soot, which protects them from degradation during transport through the atmosphere and water. Last, the Ionian Sea exhibited the lowest average pH fluxes amongst the 3 sites, and peaks in late winter, early spring at 700 meters, and summer at 4,300 meters, and predominant contribution from combustion processes, as also indicated by pH source-specific ratio values. These peaks are probably associated with atmospheric deposition events carrying both natural and anthropogenic chemical species in the marine environment. Concluding, our study reveals that PAHs in the deep layers of the northeastern Mediterranean Sea are influenced by a mixed interaction of petroleum sources and of degraded petroleum products, unburned fossil fuels, combustion processes, and transport pathways, such as maritime activities, atmospheric deposition, and freshwater inputs. Also, the spatiotemporal variability in pH fluxes highlights the significant role of biogenic and physical processes in shaping pH distribution patterns across the deep sites. Now, ongoing research on understanding and investigating the mechanisms driving the vertical pH transport in the deep layers of the northeastern Mediterranean Sea is currently being studied. So, by characterizing particulate matter, we can understand and examine how different forms of carbon influence pH transport, linking biomarkers to biogenic fluxes and carbon cycling processes. So here you can see the temporal variation of some bulk parameters like organic carbon, carbonates, and opal. Now, higher fluxes for these elements indicate processes that accelerate the export of all biogenic constituents, while mass flux is the key factor controlling organic carbon flux and consequently all biogenic fluxes. So by understanding the significant role of biogenic fluxes in the downward transport on PAHs is crucial. And let me close with some acknowledgements. Sediment traps samples were collected in the frame of the Greek National Project CRIPIS for integrated observatories in the Greek seas. And this study was financially supported by the project NAV Green. Thank you very much.
Thank you, Ms. Skilaki, for the very nice presentation and for keeping the time also. So questions from the audience? Ξανοίξε, το ανοιχτό είναι.
Okay. Yeah, thank you for your presentation. I guess you answered my question at the end with the last slides when you showed the change in the particle flux.
Yes, yes.
Because I was wondering during your presentation why there was this change in the pH fluxes with depths, but apparently it's linked to the change in the particle flux.
Yeah.
So the concentration should stay about the same throughout the water column.
Okay.
Or do you know if they change or not?
Yes. Can I see my presentation so I can go to the slide? Ναι. Εγώ το κλείσα. So you are talking about this slide, and
yes, there you see that fluxes are decreasing with depths.
Yeah, actually, for instance, in the Ionian Sea, we can see that seasonal changes and extreme events like possible dust inputs are influencing pH fluxes, enhancing productivity and organic matter export to the region. Also, we can say that higher fluxes that observed in the upper water column maybe they are due to upwelling of intermediate waters that they enriches with nutrients the euphotic layer, thus increasing productivity and organic matter export. Or in the bottom trap, if you have highest increased fluxes, except for vertical transport, you can see lateral transport. So for instance, in the Ionian Sea, deep water formation masses can transport from the Adriatic Sea by bottom arrested currents to the abyssal layers of the Ionian Basin. So we can see in relation to proximity on point sources, or if it is influenced in the bottom trap or in the upper.
Okay.
And also, yes, and here are the concentration of PAHs, because I didn't show them, I only showed the fluxes. But we observed that we have like not a lot of changes in the concentrations. Like in the fluxes, in the North Aegean, we have 4 to 5 times higher. But in the concentrations are all in yes, 1 order of magnitude. Okay. I don't know if I answered.
Now we can discuss about that later on. I was wondering if there was a change in the concentration with the water depths that could indicate some other processes, like a lateral transport or some transformation.
It depends in the load of particles rather than the carrier. Maybe.
The pH. Yeah. Well, yeah, but we'll see that later.
Thank you, Xavier. Did the right question. Okay.
Do we have another question? Okay, thank you, Ms. Kilaki, for the excellent presentations. Okay, and we're moving on to the next presentation with the title Flood Risk Assessment of 2 Greek island ports under the impact of rising sea levels, delivered by Mr. Hadjistratis. Co-authors Monioudi, Halazas, Papaioannou, Moschopoulos, Velegrakis, and Polioropoulou.
Good morning, everybody. I am a PhD candidate here at the University of the Aegean at the Laboratory of Coastal Morphodynamics and Marine Geology. This work will present some results about the assessment of flood risk on 2 Greek ports under the impact of climate variability and change. And it has been done for the project RESPORTS, Enhancing Resilience on Greek Ports. So first of all, ports are of high economic importance because they form crucial nodes in the supply network. About 80% of the global merchandise trade passes through ports, while in the case of small islands, they are usually the only location, the only entrance for the supply of local communities with basic goods such as energy and foods. However, at the same time, they remain quite exposed to marine hazards, meaning the wave action, also storm surges, and the rising mean sea level due to climate change. So this can, of course, induce a lot of adverse effects on the functionality of the harbors. It can cause also terminal and vessel damaging, and it can cause substantial disruptions and delays on the supply chains. These phenomenon, of course, are projected to be increased both in magnitude and frequency in the future under the impact of climate change. So now to give the definition of extreme sea levels. Extreme sea levels are defined as the summation of relative rise of mean sea level due to ice melting induced by climate variability and change. There's also the astronomical tide, which in the, as you know, in the Aegean Sea is relatively negligible. And above of that, we have also the episodic extremes during extreme events, which is the meteorological tide, the storm surge, and also the wave action, which is in our case is approximated as 20% of the offshore significant wave height. There is a database which is described by Vzdoukas et al. 2018 with projections of extreme sea levels along the global coastline with a resolution of 25 kilometers. These projections of extreme sea levels concern 2 climatic scenarios, one moderate and one pessimistic scenarios of concentration of greenhouse gases in the atmosphere for 2 reference years, 2050 and 2100, and for various return period events. So the objective of this work is to study the exposure to flood risk under the impact of climate variability and change at 2 island ports in the North Aegean Sea. So here are our study area. The first port is the main port of Chios Island, which connects Chios with also the mainland of Greece. Chios port has about on 2022 it had about 150,000 arrivals. So it's the most important port of the island. And the second port is Pythagorean port in Samos Island. It's the second most important port in the island, connecting mainly Samos with other touristic destinations in South Aegean, like Kos and Rhodos. Now, in order to simulate, to predict the flood risk, we performed some hydrodynamic simulations using the least flood FP model. This model runs in 2D on a structured grid and models the water flow on the ground, taking into account differences on the slope and also the soil friction. This model has been initially designed for assessment of river floods, but has recently been used also on coastal flood risk applications, including an application on the design of a pan-European early warning system for coastal flooding, but also on assessment of flood risk on ports in Caribbean. This work has been done by Beauvais et al. 2020. About the model, the hydrodynamic forces at the boundaries of the model was the extreme sea level projections retrieved from the database that I was referred in the previous slide. We consider only the pessimistic scenario. We show results here only for the pessimistic scenario. And the extreme sea level for this case was 1.2 meters for 2050 and 1.65 for 2100, considering the extreme event with 1 in 100 year return period. Simulation duration was set to 10 hours. Quite important also, we used quite high resolution digital elevation model available from the Greek cadastre. There are, of course, some issues in this model on the vertical accuracies, but it was by far the best choice we had for the simulations. While the friction coefficient was retrieved from the coastal zone land use land cover dataset from Copernicus, and then using examples from the literature, we assigned the different values to the Manning coefficient. So first, the results about the Chios port. We had the two flood maps. The different colors represent the flood depth along the study area. As we go more to the yellow and red colors, the depth increases. So you can see that on the north side, which is where is the main dock of the port, the whole dock is totally flooded, and the flood is propagates even more inshore on the residential areas. Also on the southern part, where some yachts are berthed usually, we have quite some, we have a high extent of flooding. The flood there actually is the maximum extent, reaching to 165 meters. While also the coastal road that connects the north and the south parts is also totally flooded. And this is important also because along this road there are a lot of restaurants and other touristic businesses. Then the things get even worse on 2,100. The total flooded area is almost doubled. The maximum extent is 180 meters. We can see now that the flood is not only the ports, but it has propagated substantially also on the residential areas, both on the north, on the south, and also on the central parts of the city. Now about Pythagorean port in Samos. There is flood in a lesser extent. The flood reached on the residential areas and not to as a big extent as in Chios, but still it propagates about 55 meters behind the port. We can see actually that the main dock on the south part is also totally flooded. And the things, of course, get a bit more worse on 2,100. Max flood extent reaches up to 65 meters. There is some inundation also on the coastal roads of the city. And this, of course, shows that not only the port going to have some functionality problems, but there are also issues for the safety of for the damages on the local community. So to wrap up, first of all, least flood seems to can support flood risk assessment on ports quite efficiently. One important issue of the current stand is that we used high-resolution topographic data. As I said, of course, there are some issues with vertical accuracies. So we propose that the usage of even better high-resolution data retrieved mainly maybe from LiDAR surveys. It can be very beneficial on flood risk assessment in the future. And of course, we saw that both ports are highly exposed to flood risk. We expect adverse consequences both for port functionalities, but also for the local communities. And of course, this shows that it is urgent to take adaptation measures and to build the port resiliency on both locations. These are the references that were used in these presentations. Thank you very much for your attention.
Thank you, Mr. Handzi-Stratis.
Yeah.
Okay, questions from the audience?
Congratulations, Mr. Handzi-Stratis. It's a very interesting work. I would like to explain to me a little bit about the setup of the least flood model, the boundary conditions, the time scale that you have used.
Yeah.
Thank you.
So basically, can I use the presentation again? Okay. Yeah, so basically, the boundaries are set all along the docks of the port. So we assume actually that all the extreme sea level will, including also the waves, will pass through the entrance of the port and will approach the docks of the port and also the coastal road. So actually, sometimes we run also a more conservative scenario where we consider that the waves are completely attenuated by the breakwaters. So actually, the best, let's say, solution is to run one more conservative model without the wave action. And this simulation that I show here, that we assume that all the waves have passed through the entrance to the harbor. because apart from the waves, we have the rising mean sea level that it cannot be stopped by the breakwater, and also we have the storm surge that again gonna pass through the entrance. About the duration, you said we set a duration of 10 hours. We found in yeah. No, it's not. No, we set at 10 hours. We found this is an indicative duration of storms in the Mediterranean. I can show you later the reference if you want to discuss further. And yeah, the Manning coefficient, as I said, it was retrieved by the land cover dataset of Copernicus. And of course, it was adjusted to the same resolution as the digital elevation model. And yeah, basically, that's the setup of the model.
Do we have another question? Yes, Yanis.
Congratulations, very nice work and presentation. And it's a very important effort in local areas here in Greece, and not only, but it's good to do that in as many areas as we can.
Yeah.
And it's good that you're using the high resolution DEM from Cadastre. But as you said, there are several uncertainties depending the area you are. Have you thought any actions that field observations or to how you can improve in an area like this these uncertainties?
The most
because it's very important
component. Of course. The most obvious one, of course, is to do some topographic surveys there, or to because actually we are talking about ports that you expect actually to have at least one digital elevation model, at least at the harbor area. And yeah, apart from that, I think it's good if LiDAR surveys start, let's say, to be increased in Greece in general, at least for ports that are critical infrastructures. And it's also a legal obligation for each EU country to assess flood risk on ports.
Yes, Adoni, go ahead.
Yes, hello. Good morning to everybody as well. And that was a good presentation, Mr. Hadisakis. And of course, Professor Doulakis has, it's absolutely right. There is a big problem there, as very well known, that we don't have enough information to be able to identify the mechanism first, and secondly, the extent of this type of floods. That's, of course, the first and most rough estimation. Because then, of course, we will need to have some other kind of approach what happens with the waves. When they go inside, we have boulders within the port basin and so on and so on. And finally, we'll be able to do that. But the most important thing, as you mentioned, and Yannis mentioned as well, is that we do not have enough information for the bathymetry and for the elevation, which is even more important. This is something that we should certainly upgrade. But one thing I want to say here is that it's not just a Greek a Greek malaise. Basically, it happens all around Europe. I know Mr. said this, that we don't have this kind of information along the European coastline, and I think this has starting to become a little bit peculiar. Because, for example, the Barbados Island, which is a smaller Caribbean island, has done that 20 years ago. And this is not acceptable in along our coast. That's what I want to say. Thank you very much.
The last one.
Congratulations for the work. What do you think, from an engineering point of view or a construction point of view, can the states or the local authorities can do to prevent such scenarios? Is anything feasible or anything possible to do to prevent it or confront it in the future?
Okay. I'm not a specialist at port engineering, okay, but as far as I know, the best action is just to increase the height of the docks, but then this has, of course, some adverse effects for the local communities because all the view at the coastal road that gives all the added value on, you know, the restaurants and the cafes and stuff, going to be decreased, of course, and I think that people do not want this solution. That's the problem.
Thank you, Mr. Hadjistratis, for the excellent presentation.
So we're moving on to the next presentation, entitled Enhancing Bathymetric Position Index, BPI, a key solution for benthic habitat mapping in areas of complex microrelief, presented by Mr. Karsiotis.
Good morning. My name is Panagiotis Karsiotis, and my affiliation is the University of the Aegean. In this presentation, I'm going to demonstrate how some simple transformations of the bathymetric position index, of the BPI index, can significantly increase the accuracy of the semi-automated benthic habitat mapping techniques. Okay, needs more pressing. Benthic habitat mapping is traditionally done by an expert manually. However, nowadays, several automated and semi-automated techniques have been developed, some of which are simple to use, while others more complicated and demand some expertise too. Benthic Terrain Modeler, BTM, is an easy-to-use add-on toolbox for ArcGIS that is used for creating bathymetric derivatives, as well as it is used for supervised seafloor classification. For the seafloor classification, it uses the bathymetry, the slope, and the BPI index, as well as a classification dictionary in order to divide the seafloor into geomorphological units. In our research, we used the mapping results of the BTM, as well as the results of the manual mapping, and compare them in order to estimate the advantages and the disadvantages of such easy-to-use toolboxes in habitat mapping. Our survey area is the Gulf of Gera here in Lesbos Island, where previous research by Manoutsoglu et al. Revealed the presence of thousands of biogenic reefs that consisted mainly by assemblages of dead and alive bivalves and gastropods. These assemblages create the complex microrelief of the seafloor of the Gulf of Gera. In order to map this microrelief, we collected high-resolution bathymetric data, as well as backscatter data with a multi-beam echo sounder. And after a pre-process of the bathymetric data in the software of PDS, we created the maps of the bathymetry and the backscatter in ArcGIS. Then we proceeded to the manual mapping. We used the slope and the curvature in order to delineate the reefs. And in areas of very complex relief, we used also the backscatter, as well as bathymetric profiles and the grid of bathymetry with increased contrast in order to map the reefs with the highest accuracy possible. After we mapped the reefs, we calculated their geomorphometric characteristics. Then we proceeded to the manual mapping, to the semi-automated mapping, and we used BTM with its conventional methodology. However, BTM failed to properly map the reefs. in order to, the reason behind this failure was the very small relief and the complexity of the seafloor. In order to overcome this failure, we proceeded to some transformations of the bathymetry and slope, and then recalculated the BPI index. In a first attempt, BTM before S2, we used this expression over here. The first term discretizes the reef's peaks, while the second term discretizes better the perimeter and the slope of the reefs. We mapped again the reefs and again extracted the reef's geomorphometric characteristics. In a second attempt, BTM before, we used just this expression, but with different values in the classification dictionary, and again mapped the reefs. Then we continued to comparing the results of the mapping. For the shape comparison, we used the goodness of fit index that measures the percentage of overlap between the polygons of one map in comparison with the polygons of a reference map. After using this equation, we end up with a unique GOF value for each polygon of the reference map, and then we create the map curves of these unique values. The area below the graph, below the map curve, gives us a unique GOF value for the whole map. It's the mean GOF value for the whole map. When it goes for the detection accuracy, we used the indexes precision, recall, and FNA score that gives us a meter of the correctly mapped reefs in comparison with the incorrectly mapped reefs. After following this methodology, we ended up with 3 maps: the manual mapping, the BTM before S2, and the BTM before. As you can see in the table, the number of the reefs detected by BTM before S2 was the highest, followed by BTM before, and then by manual mapping. This happens because semi-automated techniques indeed did detect more reefs. However, it also occurred due to the phenomenon of fragmentation. Reefs that the expert mapped as one large reef, the semi-automated techniques tended to fragment them and map them like several smaller reefs. That's also the reason why the manual mapping has detected reefs of bigger area, perimeter, and height. When it goes for the seafloor coverage, we notice that manual mapping and BTM before S2 has the same percent, while BTM before has a smaller percent of seafloor coverage by reefs. The reason behind this difference is the fact that BTM before detects reefs of smaller area. It does not map the slopes of the reefs as well as the BTM before S2. When it goes for the detection accuracy, we notice that all the indexes have high values. This means high accuracy in the detection. However, indeed, there are some reefs detected especially by the semi-automated techniques that have not been detected by manually by the expert. And these reefs tend to be found to occur on the perimeter of the Gulf, where the water is shallow and the relief of the seafloor is very complex and small. However, these reefs do follow the patterns of distribution and shape of the Gulf of Jera as they are proposed by Manuccioglu et al. And therefore, it is very probable that they are correctly mapped. The expert failed to map them manually. Now, for the shape comparison, we see again that the GOF values are high. Here you can see some reefs that have a mean GOF value. And you can see that the accuracy is high, while reefs with max GOF value tend to coincide with the results of the manual mapping. Minimum values are observed in cases where some reefs have not been detected by the semi-automated techniques. However, there is a minimal overlap with neighboring reefs manually mapped, as you can see here and here. A second case where minimal values occurred were in areas where this kind of errors were present. These errors tend to coincide with areas where the route of the vessel overlapped, and it's due to errors in the bathymetry. in the bathymetric grid. In conclusion, BTM is it's very easy to use, and as expected, it's very quick and easy to map semi-automatically, supervised the reefs, the micro relief of a seafloor. In some cases, it's also the results of the semi-automated mapping is higher. The accuracy is higher than the manual. However, high-quality bathymetric data is needed, and therefore, a very careful data collection and data pre-process is necessary. In cases where these errors cannot be eliminated, some post-mapping corrections might be also needed. Finally, I would like to point out that transformations of the BPI index, as those presented today, can take several forms, and many modifications and transformations can be made in respect always with the characteristics of each seafloor and its environment of the surveying area. Thank you.
Thank you, Mr. Karsiotis, for your very interesting presentation. Are there any questions from the audience?
Thank you very much for your beautiful presentation. I was just wondering, just to get it clear in my mind, so you went and you had your perfect bathymetry, let's say perfect, as good as it gets with the MBS, and then you wanted basically to classify the sea bottom, correct?
We knew that there were these reefs, and we wanted to delineate these reefs with the highest possible accuracy.
Perfect. So my question is, after you did all this process, did you have any ground truthing? Did you go back to check about your product,
yes,
your accuracy?
Show you. The ground truthing has already been done by the research of Eva Manuccioglu. There were photos and videos by divers, so we indeed knew what was there. But you mean ground truthing about the shape of the reefs? No, there was no ground truthing. We more or less blindly trusted the manual mapping, and the high-resolution bathymetric data.
In any case, it's a really good work. Congratulations.
Thank you.
Maybe next time.
Hello. Okay. First of all, very nice paper. It is, of course, a methodological paper. And the question I'm asking now is, how do you plan, after you have this kind of formation, how do you plan to use it and for what purpose?
Okay. First of all, you can use this data, the mapping data, in order for spatial planning. For fisheries, I mean, in Gulf of Jerry, it's not such a big problem, fisheries, but in gulfs like Kaloni, where there is high fishery pressure, many legislation, you can use this planning for spatial planning, where you can fish, where you cannot. Maybe in some cases with photographometry, you can make some assessments about the diversity, about the habitats, and if you can correlate the habitats with the fisheries, you can make estimations about the ecology of the area. but also by a geological point of view. You map the seafloor view. You see what there is down there quickly, fast. Yes.
Can you
data in the coastal areas to be able to, proper calibrations, proper validations, proper. That's what we're seeking. But anyway, it's a good first start, to start becoming more accurate for the mapping of this type of environments. Okay, thank you.
Thank you.
And I guess you are going to test the methodology for with data from the Gulf of Kaloni.
There might be some,
okay,
maybe this kind of techniques or maybe other mapping toolboxes.
Okay, is there any other question? Okay, thank you, Mr. Karsiotis.
Thank you.
Okay, in the next presentation has the title Marine Habitat Mapping and Shallow Geomorphology Around Yali Island by Ms. Jafaridou, Petsimeris, Ikonomou, Chasiotis, Andreadis, Poulos. The presenter, Ms. Jafaridou. Thank you.
Hello, everyone. My name is Zafarid Vasiliki, and I would like to present to you our work about the marine habitat mapping and shallow geomorphology around Yali Island. Okay. Yali Island is a small, uninhabited and volcanic island located between Kos and Nisiros in the Southeast Aegean Sea. Two companies actively extract pumice and perlite on land, and the wider area also belongs to the Natura 2000 network, as we can see in the map here. Despite the area belonging to the Natura 2000 network, the marine environment of Yali has not been the focus of any study in the past, so there are no available marine habitat data, except from submarine hydrothermal seeps in very shallow waters. The aim of this study was the investigation of shallow geomorphology and marine habitat distribution, as well as the assessment of potential anthropogenic pressures due to the onshore extracting activities. November 2023, a dense grid of parallel and vertical survey lines were conducted with about 80 to 100 meters distance between each other, covering 6 square kilometers. About 250,000 bathymetric points were collected and filtered, 41 sediment samples and 82 videos were collected as well. 2 echo sounders were used for bathymetric and morphological data collection. One of them was a multiparametric sonar, which was used for both bathymetry and morphology while operating in different frequencies. And it was also used as a navigation system. A GoPro camera was used to obtain the ground truthing videos, and then the data was analyzed using SonarWiz and QGIS. Automated classification was tested. However, because of the complexity of the area, it did not produce satisfactory results. So eventually the mapping was performed manually. The sediments were collected using a Van Veen grab, Out of the 41 total samples, 37 consisted of coarse sediments like sand and gravels, and the other 4 consisted exclusively of seagrass, so they were not analyzed. The samples were dry sieved in the laboratory, and then they were analyzed using the GradStat software in order to obtain granulometric parameters like mean size and standard deviation. The survey extended to a maximum depth of 67 meters, and it showed uneven morphology and microrelief throughout the area, as well as an extensive reef to the south. The slope ranges were quite wide, with smoother increases observed in shallow waters, as well as in waters deeper than 50 meters. In the majority of the area, slopes increased up to 45%, with local outcrops up to 80%. And the reef itself showed smoother increases in slope. Another important characteristic which was observed in the area were the irregular and extensive reflections throughout the water column, as they were observed in the bathymetric records, the side scan records, and also in a very small number of videos. Those were attributed to hydrothermal fluid venting, which occurred between seagrass meadows, rocky relief, as well as sand. There were numerous isolated seepages throughout the area, as well as 3 extended areas which were detected, the deepest of which was to the north, between 25 and 55 meters. The others were to the south and to the southeast in much shallower waters. Similar but ambiguous reflections were also detected in shallow waters, but they couldn't be classified. The reflectivity and appearance differences were attributed to different textural differences and variations. Those were attributed to sandy substrates, sand ripples, gravels, and rhodoliths, then seagrass, rocky relief, as well as a 6th transitional type, which was scattered rocks and sand. Here we can see the spatial distribution of the observed habitats, where we can see the lack of seagrass in front of the port facilities here, as well as the rhodolithic fields, which extended in the deeper limit of the area of study. All the reflectivity types, except from the transitional one, were corresponded and were classified according to the network of protected areas, Natura 2000, as we can see in the map. For the sand and the sand ripples, the description notes that they're found in shallow waters. However, for the present study, they were also found in deeper waters, which is why the general code 119 was used here as well. Additionally, for RT4, Kimodotsia was also present locally instead of only Posidonia. A few anthropogenic targets like moorings, debris, and construction bases were found to the east and near the port facilities, mainly within sand. A small number of anchor scars run through seagrass meadows, and a linear target which corresponded with a telecommunications cable was detected to the north with a length of 230 meters. The mean size obtained from the sediments helped verify the side scan reflectivity types. Samples were obtained for only the 3 first reflectivity types and not seagrass or rocky relief. For the sand, while it appeared visually homogeneous, it did include various percentages of gravel, which gave it quite a wide mean size range. That wide range could also be due to the lower specific gravity of pumice, as well as the placement of the sonar on the boat and the depth variations, both of which could potentially affect reflectivity. Gravel and rhodoliths, as well as the sand ripples, had narrower ranges. The other granulometric parameters also had wide ranges, which could indicate the mixing of various sediment subclasses of different origins. In conclusion, the seabed around Yali Island presents uneven morphology like outcrops, reefs, steep slopes, as well as submarine fluid seepages, all of which are typical of a volcanic environment. Of the submarine fluid seepages observed here, only one area corresponded with previously mapped seepages. All of the 5 main habitats, which were distinguished and categorized according to Directive 9243, are typical in the Greek shelf, And additionally, Posidonia oceanica and rhodolithic fields are classified as priority habitats according to this directive. A few human imprints were identified. However, there was also no evidence of considerable pressures stemming from the onshore extraction activities. Yet biodiversity and other parameters need to be examined as well. Thank you very much for your attention.
Thank you, Ms. Japaridi, for the excellent presentation. Questions from the audience now. Yes, please.
Thank you for your presentation. It was perfect, by the book, everything. I was just wondering, at some point you said that there was a Cymodocea nodosa present. And I was just wondering if you got to see this with your sonar, with your camera at some point.
It was visible in the available videos. In some of them, in some areas where we saw Posidonia, alongside it, there was also some Cymodocea nodosa.
Did you do any effort to map those meadows? Draw a polygon, or it wasn't that easy?
Not really, because it wasn't visible from the sonar at all. It was only visible through the videos, so mapping it could be difficult.
Okay, thank you very much. Congratulations again.
Thank you.
I was going to ask if there is one more question, or two maybe. Yeah, well, Andoni, of course. Thank you.
Yes, hello. Yes. Actually, what the previous member of the audience asked, it's exactly that everything is done by the book, for sure. Then the last, we got some kind of conclusion. Didn't identify any kind of human interference, none. In an area of so big mining activities, we didn't identify any kind of, let's say, some trailings or tailings in the water or something like that.
The extraction itself takes place on land, but from what we saw from the appearance and the distribution of the habitats, there wasn't any pressures that we recognized. In front of the port, yes, the seagrass wasn't there, but that's the case for most ports. So.
Okay.
Okay. Is this part of a larger project? I mean, is this research part of a larger project? I mean, are you mapping other? Are you involved in mapping other?
It was just for this area. Then,
Okay, so there's a database being built. Okay, this is okay. Okay. Thank you. Thank you very much.
Thank you.
Okay, and for the last presentation of the session, we have a presentation with the title "Changes in Prokaryoplankton and Nanoplankton Community Structure After Artificial Dispersal." The authors are Alexiou, Arvanitis, Polidoros, Tsengergi, Voudanda, Karayani, Kormas, Meziti. It's going to be delivered by Mr. Arvanitis. Thank you.
Hello. So, good morning. I'm Philippos Arvanitis. I realize now that this presentation might be a bit irrelevant to the previous ones. Anyway, I'm here today to present to you our results from our study, which is a part of our bachelor thesis, which we performed collectively with Alexiou and Polydorus in the back. So, for this study, we collected surface samples from 3 different coastal areas, which you can all see in the map. 2 of them from the Mediterranean, 1 from the Atlantic Ocean. The samples were inoculated at the same medium and were incubated at the same conditions. Our basic questions for this study was to search how do prokaryotic and heterotrophic nanoflagellates chain assemblages change over time, and to find out if it is true that everything is everywhere and that the environment selects. So water surface samples were collected from the Bagnol site and were filtered and later autoclaved. The water was used as the growth medium for all our experiments, for all our treatments. The inocula that were collected from the 3 different sites were filtered in order to keep at the end all the prokaryotic and heterotrophic nanoflagellate populations, using filters that were to keep microorganisms that were smaller than 11 micrometers. And I should also say that all our experiments were prepared in triplicates and incubated at 18 degrees Celsius in the dark. Water samples were collected daily and were used for epifluorescence microscopy and flow cytometry analysis for cell counting. Water was also filtered in order to perform DNA extraction experiments. For the DNA extractions, specifically, we sequenced with Illumina using the known prior 16S for prokaryotes and 18S for eukaryotes RNA genes. The results were sequenced using a specific software and analyzed and clustered at 97% similarity threshold in order to obtain our OTUs, which, as you can see, stands for operational taxonomic units. Some of the questions we had for the study are: how do microbial communities change over time? Do we see any communities divergence through time? And are there any common groups that persist in all treatments? So these are the results. Here you can see the results for our cell counts experiments for bacteria and heterotrophic nanoflagellates. As you can see, we had typical growth curves for both bacteria and HNFs. And based on these curves, we decided when we should take sample for DNA sequences. So the sampling began at T0, which is the start of the experiment, T2, which is the end of the exponential growth phase for bacteria, and T4 and 5, which is the start and the middle of the death phase accordingly. Here are the Ray-Curtis similarities between samples for prokaryotic in the upper triangle and eukaryotic communities in the lower triangle. I realize this table might be a bit chaotic, but I'll try to explain it as best as I can. So for eukaryotes, we saw similarities between time points of the same treatment that were very low and usually under 10%, apart from the samples from Bagnols that after T2 seem to increase after larger anyway than 20%. The inoculum samples were increased only between parasiticos and Bagnols and reached 33%. You can see it with the red numbers. And finally, similarities between different treatments at the same time points seem to be very low. That you can see with green. Moving on, for eukaryotes, similarities between time points at the same treatment seem to increase over time. These are the underlying numbers. While similarities between treatments here also seemed to increase, but after T4. And finally, higher convergence was compared to prokaryotes. Here are the taxonomic results in Here are the taxonomic results. This is the favorite diagram of our table from our teacher. You can see that there is a prevalence of archaea in all samples at T0 that seemed to decrease later on as the experiment went on. Here are the results for the taxonomy for prokaryotes at the phylum level, which seemed to, here we saw an increase of differences between inocula and T0, but from T2 and later, what's interesting is that we saw a prevalence of gamma proteobacteria to all our samples, which seemed to be absent in all initial time points. For eukaryotes now, there was a different of compositions at all time points and sites. We observed in all our samples a mix of saprophytes, mixotrophs, graziers, and autotrophs. At T4 and T5, in more detail, we saw that parasites and saprophytes were very high in all our sites. And finally, what was intriguing to us was that the relative abundances of potential grazers was lower than expected. So these are the taxonomic results from our taxonomic analysis for prokaryotes at genus level. You can see that all the genera that were present are above 1% in at least one sample. This is an logarithmic scale, so with the orange colors, you can see that the relative abundance is above 1%, while with the boxes with the red colors seem to have relative abundances over 10%. So at the beginning, we saw a high number of unclassified OTUs, particularly for archaea, which indicates that there are still a lot of new species and unknown diversity in the marine ecosystems that we do not know about. It also highlighted the importance of Candidatus nitrosopumilus, which is right here. That showed us also the importance of ammonia oxidation of phototrophic growth in all inocula. Moving on, at Parasiticos and Woods Hole inocula, we saw a great increase of flavobacteria, which also showed the importance of degradation of complex organic compounds and particulate organic matter. For binules, Rhodobacterales seem to increase after TIN2, mainly that site. Rhodobacterales is a widespread marine organism, which is well known for its metabolic versatility in low nutrient conditions, especially such as that of the Mediterranean Sea. But perhaps our most important finding, as also mentioned previously, was the prevalence of gamma proteobacteria, and mainly the order Alteromonadales, which showed an increase after T2 on all our sites. Alteromonadales are known for their rapid growth on diverse organic compound environments. So finally, we set some questions at the beginning of the presentation. I hope you did not forget. First of all, how do microbial communities change over time? Changes in our communities seem to appear after T0 at all cases. And we saw a shift to more flexible in terms of metabolism microorganisms. The Candidatus nitrosopumilus was the only one that was stable in our communities. Also, do we see any communities convergence through time? We did not detect a significant communities convergence through time, at least at an OTU level, but we saw a convergence on metabolic functions. They seem to use alternative and more complex organic compounds for the rapid to increase their growth rates. For eukaryotes now, convergence on trophic groups there seem to be a convergence on trophic groups such as parasites and saprophytes. That indicates the prevalence of dissolved organic matter concentrations related groups during bacterial death phase. So, in conclusion, the environment does not select, probably, but it seems to prefer some microorganisms with certain functions. At this point, I would like to thank the staff of the OB for hosting us, and Susanne Thomas for sampling and shipping the inoculum from Woods Hole. And you for your attention. Thank you very much.
Okay, thank you, Mr. Arvanitis. Now let's take questions from the audience. We have a question. Okay, let me ask one. In a slide, you show a table with the Brigg-Curtis similarities, right?
Yes.
Yeah, okay. So did you try any cluster analysis or MDS analysis so that we can show the results in graphs rather than tables?
Yes, we have made an MDS and other
analysis.
Yes, which I do not know how you call in English. But they were not ready at the time when we had to submit our abstract, which was this was a part of our thesis, and we haven't came to a conclusion since then. So they were not ready, and we decided not to show them in the presentation.
So I suggest you proceed with it.
Yeah,
probably. Okay. Any other questions? Yes, Adoni. Thank you.
Yes, thank you. Obviously, a lot of work. Two things. Basically, I didn't understand. The 2013 samples were from Woody Woods Hole. It was taken in 2013?
Yes.
And the others?
All of the samples were from 2013. We made the analysis.
Okay. And what was not very clear to me, so basically, you have samples in Mediterranean, samples in the Eastern and the Western Atlantic. Okay. So what do they show? Do they show any particular difference or everywhere is the same? Maybe I didn't understand. You explained that I didn't understand it.
No, there were a lot of differences between all our samples. I guess the greatest similarities were shown between, there were two levels for prokaryotes and eukaryotes. Eukaryotes showed a lot of differences, but for prokaryotes, we did not see that. Actually, for Banyuls and Parasiticos, they seem to have more in common than from
Woods Hole.
Woods Hole, yeah.
Yeah? So basically, Parasiticos and Atlantic has got the same characteristics, not the Eastern and Western. Interesting.
Yeah.
What do you think is that?
I'm not sure, to be honest.
Okay.
Miss Mezziti. Yeah.
Yeah, that seems good.
Okay, do we have any other questions? These here are the other members of the group of the. Oh, okay. Okay. If we don't have any other questions, I think we have reached the end of this session. So let's call it a day and get some coffee, I think. Okay, so great. Thank you.
Ναι, ναι, ανοιχτό. Good morning, everybody. Thank you for your participated to this excellent keynote speaker from Professor Khalid Filic. Also, I want to welcome the people are in virtual mode. Before I call Dr. Khalid to the panel, I would like to mention some few things about the professor of Khalid Filic from the Department of Theology in Faculty University in Turkey, from Ege University. His academic profile pertains the study on ichthyology, fisheries, and made significant contribution to the department of Ege University, earning recognition of his outstanding achievements, promoted to the position of assistant later, and also there is a recognition of his leadership skills and expertise working in this department since 2021. His interests are on the distribution of native invasive fish species, interaction between fisheries and fisher community, fisheries biology, fish diversity and conservation issues, as well as also fish systematic and distribution. So I will be very honored to ask for Dr. Khalid Filic to come to the panel to present his prolific presentation. Dr. Khalid.
Kalimera. Kalimera. Allow me to briefly introduce myself. Again, I'm Halit Filic from the Faculty of Fisheries at Muğla Koçman University. As mentioned, Dimitri, my concern is fish and any research providing data about the fish and fisheries. By the way, I would like to thank everyone, firstly, who made this meeting possible for their dedicated efforts. Find the chairman, dear Michael, for providing me with this valuable opportunity. About the presentation, there is a big picture in front of me, and it's a difficult task, but there are many issues that need to be discussed. Therefore, I have to plan very well where I will start and where I will connect the topic. So let's start. It is said that no wind will be good for those who don't know where is the port they are going to. So I am an academician who loves only science and try to share what I have learned. I love science because it's alive, constantly changing and evolving, and for more important, open to discussion as we will day to day. Question I've been asked most lately is, where is the fish? I tell them in order to understand what's happening to the fish, we need to find the starting point and proceed step by step. Don't be afraid, we want to go back so far, but we need to find the starting point. In a book called How Man Became a Giant, the authors said that a mythological saying, there is a giant in the world. And continue, he has hands that can lift a locomotive without at least effort. He has feet that can travel thousands of miles in a single day. He has wings that carry him up above the clouds higher than a bird fly. He has such fins that he can swim on top of the water and under the water better than any fish, etc., etc. And they end up like this. He makes the world over to suit himself, plants forest, unity sea, water deserts. What a true statement, in my opinion, to start the story with. Arguably, mythology began with worship of ancestors by putting our ancestors on a pedestal, smoothing out in perpetuation and difficulties that are a part of anyone and everyone. In Greek mythology, giants are extraordinarily large, strong, and often terrifying mythological creatures. But unfortunately, the giant here is this era is not a mythological, it is real. The giant is us, our species. We can say that the first step of our damage, the environment, began with the first settled communities, the first transition to settled life. Imagine a hunter-gatherer community that spent 6 months in a one location, then moves to another area for another 6 months. They are not worried about stocking anything. Their only purpose is to survive for another day, just food and water, that's all. However, with the transition to settled life, we see that humanity has evolved and started storing more and more things. This is the overkill hypothesis, maybe you know. There is a great harmony between human migrations and the extinction of large animals around the world. Wherever you go, the story is the same. You imagine we did this only the very basic spears and arrows. Imagine what we are doing with what we have today. At the root of each problem is human overpopulation and overconsumption. Overpopulation is particularly destructive to also aquatic ecosystems and to fishes because humans are concentrated on the rivers and estuaries. And now humanity is an organism that began to affect the earth by the major geological phenomena you do. The earth has abandoned geological scale. Now it's changing at human speed. This is not our starting point either, but there are very striking figures. And in this slide, I want to understand that what happened, in what period or time course, how did humanity come from the domination of the nature to the point where it destroying the nature, thus, in other words, destroying itself. Here is in my opinion, the starting point. You know this imagine very well. Human ego, or anthropogen ego. We cannot say, oh, the truth is before us, just opening the door. Just because the truth was shaped and evolved towards a certain level over time, so it is necessary to look at what has occurred over time. There is a dynamic process going in front of us. It's difficult to explain this, but we have to. Humanity is destroying interaction with the environment and with the nature, acting like a viral thing. Yes, human beings are already a part of nature from very beginning, and they necessarily interact with the nature because of their needs. As humanity interacts with nature, it transforms nature and also transforms itself. And regarding the nowadays problems, when anyone says the real criminal is someone else, you should not leave yourself be confused by the terminology. You know, fish and fisheries discussion are full of scientific terminology, concepts, and complex statistics. We must wrestle with aspects of fisheries, encountering words and phrases like overfishing, recovery time, and fisheries mortality. We do not feel a connection with years like future, for example, 2100 years. So these concepts are very confuse us. We are confronted by changes that are more complex than most of what our minds typically deal with. These changes surpass any of previous experience, surpass most of language and metaphors we use to navigate our reality. So today you say anyone about the fisheries problem and colossal fish stockings, it's sounds he or she as a white noise, because we only know when the thing is already happened after that. At the point of criticizing ourselves, I can say that we overlooked the industry a lot. We have to ask what industry and the industrial revolution are. What brings the industrial revolution and industrialization? This is mechanization. The translation from tool to machinery is the key. We are talking about mass production of organization and capitalism also. Capitalism, you know very well, is a system of plunder. It's a system in which the plunder is formalized, legitimate, and restrained. Changes in fisher stocks are not an urgent issue of our time. It has always been like this. But of course, marine life has its limits, and there is an ultimate ending point for species. We are in a period where this ending is getting closer and bigger. And how beautiful Rachel Carson said in her book, The Sea Around Us. We all come from sea. And yet, sadly, I am not part of the earth, and we are no longer than 70% of water. We are 90% of oil and its power. When we consider the numbers, we have some parameters. One is population growth. The Earth's population numbered 700 million when we started exploiting the coal and the oil that gave us super strength. We are now 8 billion. Having cheated the system and dug our way down to ancient sedimentary deposits, pumping down back to the surface, kindling fires. We went fire to our will. We drove the ships right in the storms to shovel up the fish. All thanks to the power of combustion. All this has increased slowly but surely for more than 2 centuries, and never more than at this very moment. In fact, the companies we call industrial fishers, for example, try to produce as much as they can their short-term interests. All these goods and products surround us now. So we need to see and point out that monster that grows as it eats and becomes more harmful as it grows. In fact, following the Industrial Revolution, we did not become a destructive species with a capitalist approach. The truth is that we have been destroying since our existence. In this very well-known diagram, the Worm and colleagues indicated that if we not take some serious how can I say? Yes. The fish stocks will be running out until the 2050. So I think we passed that too. Because we are so hypnotized by progress, our revolutions, and we lost the relation with the future, and the future is just a dream for us. And for example, for us, 50 or 100 years are like a whole eternity, a thing beyond imagination. We do not react substantial disasters in these future projections. We shrug our shoulders, and in fact, future doesn't concern us. I believe that if we put together the rapid increase in extraction habitats, industrial and agricultural pollution, overfishing and overexploitation, plus the effects of climate change, we have a real chance of the total collapse, not only fish stocks, also entire ecosystem. And maybe I may not understand who is the real culprit, but maybe the fish itself. Yeah, why not? Because they can find all my aquatic habitats. They are a source of quality food. and they can be caught in large quantities. These beautiful features have become their curse under the human rule. How difficult is to remain impartial as a human being? If it does not work for us to solve problem in our way, another point of view, the external eye must look at us. This is the being of voice of. Actually, a fisher, a traditional fisher. One of our meetings, Osman Demirkol, a traditional fisher, stayed and stand up and said that I came here to be the voice of fish, and this statement impressed me a lot because all the time I love science, I love university, I love the fisheries research, but anyway, it didn't come to my mind to be active voice of fish, for example. We are only looking all the things by the scientific way and the scientific vision. So this time was an empathy time. I had to look at all this from the perspective of fish, get out myself and try to be words of this group that could not tell us their problems. Here is the fish. I let the fish turns to talk. Evet, yes. The fish says your commercial exploitation. As a fish, as I imagine myself in the sea, the fisheries are an encircling, a basement. For fish, no place to escape, no place to hide, only net, and another net. The destruction appears on the net, a growing insidious tumor. The fisheries industry slowly swallowing the fishes. A breathtaking intimidation game begins. Those who are behind the net ask with astonishment, what's that? It is a story about fish starring fishing nets. The truth is that for decades, our fish stocks in our seas have been subjected to wild fishing without measurement of scientific basis. Our main problem is your overfishing capacity. Indeed, our fish capacity is still too much for even oceans, for the oceans. Also, current indicators based on production point to a decline in fish stocks despite increased fishing pressure. Since we do not engage in selective or ethical fishing, most of the fish are thrown back into the sea as a discard. And the fish says, you know nothing about our stocks. You have no idea how many you will catch. You do not have quota management. Everyone can fish as much as they want. The 1970s in the Turkish fisheries referred to our golden age, you know, nearly the 40 ton per boat. But let's check the situation based on this. As you can see, the number of your boats has doubled, but the catch per boat also has almost halved. The quota system will be a life vest for our fisheries and should be implemented immediately. But we learned any lessons from this? I don't think so, and a big no, because instead of reducing fishing pressures and apply a quota system, we choose to draw fishing boats right into the storms to show up fish. And the dying of fish species through the human agency is a major event. When the last fish is caught, the fishers did not know for certain that this was the last fish. The word often makes more sense in the hindsight than when people are the middle of the things. And the illegal or unreported fisheries, no matter what is done, illegal fishing has always happened and will always happen. The main reason for this is that it's easier. Because sometimes when the right pipe is narrow, it is much easier to walk on the wide one. And I have any Fisher's example for this slide, because the good manners fixed by experience is a great skill. The most beautiful narration of this slide was made by Mr. Erjüment. He's a 50 years old traditional fishers. Again, in a fishers meeting, he stand up and said, "The fish is finished. We are talking about the finished fish. We finished the fish, we caught the mother, we caught the child, we caught the grandfather, we caught and caught. So there is nothing left to lose. So let's find a way to survive from now on, because there is a generation coming after us. Let's think about them. Tiny groupers are caught and there are people who buy them. This is immoral, this is not ethical." I think these words explain this slide very well. And the fish says, "Don't catch our youngs, respect our elders." Fishing vessels, pollutants, climate change, human alteration of aquatic habitats a lot via, for example, modification of bottom types or colonization or building a dam. You know, the nature can renew and clean itself, but if you go beyond the limits and exceed the limits of nature's capacity to renew it, it will collapse obviously at some point. This is an bad example from Turkey. For example, expanding one's olive fields to feed a large family is not same as filling a pond or shore, eradicating its fish and vegetables and nesting sites in order to build on marina, road, or hotel. When we look at from where we are, it seems like a small local problem, but we encounter this destruction in every corner of the aquatic and coastline areas. We can call it complete extraction. How does our seas get polluted? Pollution, another problem of fish. It says you can count the factors of water pollution under two main headings. The first is natural resources, and the second is the water pollution caused by human activities. Pollution caused by industrial zones, agricultural activities, pollution by urbanization. You poured agrochemicals and earth soil. You poisoned our clean water sources. It says, and another problem for fish, our impacts. As we mentioned above, the human makes the world owe it to himself, but you know, the ecosystem as a whole. Even small interventions in this system, which you have a little knowledge about, so can cause minor problems. For example, you open a canal purely for economic gains, an alien fish enters, establish, and increases its population, thus become invasive. Devours everything without conscience, without local systems, without fishery economics, industry get environmental assessment also, and international conferences. The most significant constituent of the global ecology destruction is the climate change. But on the other hand, decreasing, especially the top predator fishes, is a big problem for the ecosystem. When they first arrived, the rabbit fish were welcomed as they would fill the empty harbor chain of Mediterranean. But due to over and illegal fishing, the predatory fish that will prey on these, for example, leaf fish and groupers, as such, the number of rabbit fish has increased rapidly and they became invasive. And now, when we dive any part of Gökova Bay, also Gökova Bay is a especially environmental protected area, we see nothing on the rocks, only the area like a desert because of the rabbit fishes. So under natural conditions, species are constrained by co-evolutionary processes. Species have natural parasites, predators, and competitors that control population growth, and organisms typically exploit prey taxa that have evolved defense mechanism against the predators foraging tactic. But there's two ways an alien of invasive species. When individuals of a species are introduced suddenly into an alien environment, they may find the new physical and biological factors inhospitable or even lethal. But alternatively, freedom from natural biotic control may revoke all checks on population growth. It is the liberated aliens that cause the greatest problems for many species now. And the fish says, we do not want to give up our nests. We do not want to them destroy our reproduction area. We want breathing space. But you know, for an area to be protected, it needs to serve as a national park or touristic destination. Its purpose needs to be measurable, preferably by how the national park directly delivers dividends, employment, and increased scales of goods and services in close proximity. When you discuss marine protected areas, you talk about their importance for fishing and tourism. Also, you know, when you claim to invoice to decision makers, here maybe declare a protected area, you have to use the money, economical benefits of an area. So this is very confused. And the fish says, please protect the traditional fishes because, you know, the nature is essential source of culture. The main space of it, every culture has its own shape that originates from the surrounding nature. Culture always develops within society and has its own time and process. In that process, all those myths, folk songs, or more tangible things, for example, traditional boats and fisheries, emerge as element of culture. We have to protect traditional fishes because they had a sense of respect for nature. Because of that, they knew very well where to build the ports and where to fish. A 10,000-year-old library is now burning and disappearing. This knowledge will be extinct with the death of the last generation because this is an unwritten tradition. The local community needs to understand why fishes need protection. As long as it is not understood and protected by local communities, the collapsing of the local stocks cannot be prevented. But the fishers not only be the ones blamed, they as scientists, we are also responsible for the problems we face, but we are not alone. Our higher educational policies, promotion requirements that push academics to produce fast and low quality works, politics, investors, and society's lack of interest in science, we are all responsible for the failure of scientific conservation of fishes. We could not combine knowledge, power, and state authority, nor can we ensure continuity. Decision makers must be convinced with accurate information, scientific and correct data, and a strong civil initiative. Also, decision makers must be sincere about the conservation itself. And the beginning of everything in scientific ethics, good data and quality results. Our educational systems places little emphasis on biodiversity, protection, and marine life. The country's brightest kids couldn't identify really common fish. Therefore, education on biological diversity conservation and fisheries should be included in the curriculum of primary, secondary, and high schools. And another future danger is the young scientists. Young scientists working on fisheries, especially on the specific species basis, need state support. There is a risk of not being able to transfer knowledge due to young people not choosing academic life due to future concerns, because a previous scientist has a certain age now, and a few young people are coming after us. Our methodology may be a problem. In most fisheries regulations have made to accept species conservation by adding the adjective prohibited or ban. I think this was wrong step. To find the best species practices, we should abandon trying to accept the prohibition mentality and explain methods that understand especially the local peoples and local culture and contributed their local economy. And also, as a scientist, we must do our fisheries conservation effort. Generally, that limited, very local area are not show for us and not to make our name heard in the scientific community, but also with sincerity. Thank you. Yes. Okay. And now our turn again. Thank you, fish. Fish vessels, we count, and pollutants, climate change. That's tracking down an invisible killer. Climate changes have effects on living things that weaken them. Those that are unable to adapt these effects and will disappear over time, while those can adapt can continue their lineage. But in the end, this is a difficult and critical period for both those who perish and those who survive. It was during such a period that humans emerged and took their places on the stage. In addition to climate effects that animals already had difficulty adapting to, the slow but destructive effects of us humans also put pressure on that. What can be done? There are some take-home notes suggested by FAO, which we all know well, and many of which are also expressed by the fish. So let's continue our way. Indeed, what we know about fish and fisheries biology. Most of the deductions made so far and the calculations and the equations based on them are considered to be under normal conditions, but in these days we are living not under normal conditions, so we faced extraordinary conditions. So we think about our parameters, our equations again and again, and we must try to understand features of fish more and more, because I believe that we know nothing about nature, because the nature is multi-dimensional system and we only know 1 or 2 dimensions of it. So it's very important to research and say new things about all the fish and ecosystem, for example. And to tie the subject with my title, those who love fishing or have friends who are keen on fishing know this. Generally, 3 fish sizes were caught. The fish is either small or medium-sized, which has always been caught. The biggest ones always flew away, and those big fish that escaped are told as legendary stories. It's saying that every amateur fishers know. The fish that escapes is big, the one that can't get away ends up either on a tavan table or a poor man's plate. What we all understand this, the fish then that can escape is the big one. But let's look at it from this perspective. So the fish that escapes survives and grows. Let the escaped fish not the big in stories, but really grows. And again, we mentioned about the previous slide, respect the younger ones and respect the older ones. This is what I chose this title in my presentation. And lastly, I know very, very long, but I try to shorten. Lastly, as you understand along the presentation, we have been fighting with nature since our agricultural revolution, and this is such a fighting that if we win, we lose. We as a people looking nature in our mind way, and we are trying to create the permanent landscape, but you know the nature has no constant. Change is its essence. Creation and destruction take place simultaneously in the nature. There is no separation between them. It doesn't matter where we rewind back in time. Nature is always right, has always been true and right. Creation is change. Everything is in the process of transforming in the nature. The world and the system within it are highly dynamic. These ecosystems have collapsed many times globally, and species losses have occurred in these collapses. But historical and scientific data shows that as this after collapses, species emerge again and a new biodiversity is established. My expectation of a major collapse in the Mediterranean due to human impacts, which we think could lead to species loss, is highly unlikely. Therefore, it will be more accurate to read the term collapse of Mediterranean biodiversity in the following sense. There will be a different Mediterranean than the one we are used, and it has already happened. What is the curse of Cassandra, you know? In mythology, you warn people about the disaster, but people don't listen to you, and then these things happen. We are in a phase of events where life as we know it can come to an end. We realize this phase, we are looking for solutions to get out of this phase with adaptation and new and minimal damage. When you live in a housing estate, even when you have enough money to consume marine foods or fish, you can't run away from it. Decreased water resources, ecological crisis, the danger of extinction of our planet. I am taking the first step, albeit with fear, being the voice of fish, and now I'm inside the sea. Thank you.
If there is not any questions, so you go the same again, okay, until we have the first question, okay, many times. So can we have one question? Because it's an excellent presentation. It's very important contribution. Shifting lanes, the baseline, especially in fishery science, is very precious.
Thank you,
Of course. I think Dr. Hadzefsafiou has something to mention.
Yes. First of all, to thank Professor Filis for his contribution and the acceptance of our invitation to come and join us and give us his perception on this issue of the area. And then I saw all the problems about overfishing, of the problem with the size. Even in Greece, we are also trying to reduce the number of vessels, but the pressure to the stocks is not reducing. So it's not just to reduce the vessels or the size of the vessels. We must have to change the attitude of the fishers, and we have to have in mind that we have to preserve the ecosystem and the stocks for the fishing community to still preserve and exist. And especially in our area in Mediterranean and Eastern Mediterranean, fishing communities is something that exists from the start 2,000, 3,000 years ago, and it's a part of the cultural state of the area. So it's not just preserving the fish stocks, it's that preserving our way of life and our history and background. So it's very significant to do that. And this is something that your presentation saw very clear. And the other one that also saw with the statistics and the numbers is that we know that we have problem with the fishing in the depletion of the stocks in the Mediterranean, but everyone is talking about the overfishing. But I saw that the overfishing is only 15% in reality of the problem that is that have the fish stocks, and mainly the other anthropogenic activities in the Mediterranean cause a major problem to the fish. So thank you for giving this information to the colleagues and the public. And I have that. I want you to make more comments on these two very significant issues. Thank you for my side.
Thank you.
Somebody else? No? So, regarding this issue, Khalid had a slide showing that overfishing contributes, I think, 17%, 15 or 17, but 73% of fish depletion is attributed to pollution. Okay, so why we are speaking about overexploitation since we have fishing habitat degradation by human activities? It's no logic to accuse somebody who are not his fault on that. Fishers are usually blamed on something that are also in the same part of the problem of the habitat degradation. FAO in 2001 state that fishers are not bring the problem, is a part of the problem. But they are accused fishers for overfishing because they are not well educated, they are poor, they are living in isolated places, but they are not. They are the target of the overfishing. Okay, especially in Mediterranean, in the Mediterranean, where most of the fishing activities are small scale with small intensity, both in spatiotemporal expansion. It's not logic to say that these people are are the main aspect of overexploitation. What do you think about that?
Yes, the fish is not on the blame.
Yeah. Oh, no, this is a different
definition.
No, this is a different aspect solution.
Yes.
Just because fisheries management put some dimension on legislation, but they don't ask most of them, the professionals, but also important, they don't ask about the people that do the control. Control authorities are not participate in fisheries management. So they take a law, but they're not able to apply the law. But this is something different. If you have some comment on why we are accused small-scale fishers on overfishing.
As I understood, you asking the ratio of overfishing by the local fishers. This is I agree or not agree this.
No, I'm not a bad guy. Just a comment. Okay.
In my opinion, local fishers is not responsible for the overexploitation.
Do you believe that there exists overexploitation? Because I know from European Union reports, there's no data to confirm overexploitation for the Aegean stock. There are not any stock assessment in Aegean, officially stock assessment. There's only for Hague, but European Commission states that we don't have data, but we know that the Hague stock in Western Mediterranean is overexploitation. So, as a Socratical approach, the stock in Eastern Mediterranean is also overexploitation. But for the other stock, there are not any official. Just some papers support that maybe there is overexploitation, some equations, some stock assessment models. But the official position of European Commission is not any overexploitation. There's not sufficient data to support overexploitation.
Additionally, in countries like my country, Turkey, the amateur fisheries is another problem, because lots of amateur fishers, not only the fishers, every weekend in Turkey, all the people from the 7 years old to 17 years old take the lines and go to sea for fishing. And there is no record how many amateur fishers.
From the shore. From the shore.
From the shore.
Okay.
And you can take the lines even in the Apotica in Greece.
Look,
any markets, you know.
Yes, but is there an impact? How many are the fish? How is the intensity?
No, so that's why I say we need to return science again, but ethics and quality science. We are doing, I think Thursday, some of our colleagues from Turkey, Dr. Ozan, presented a presentation about fisheries study in the Mediterranean, a comparison between a whole Mediterranean. And in one diagram, we say that the Turkey is the leader, 186 research about age and growth of fish studies, and then Greece and then Italy. But yes, there is a lot of work, but they include how many secure data? I don't know.
We don't have data.
You have, but
do we have data?
No, we are nothing about.
So we are in science that we don't have data.
We are nothing about fish, nothing about fisheries.
So just hypothesis.
But there is a big problem in front of us that I don't know what is the solution. But I am sure one thing: we get to knowledge means academia, and the power means the locals and the NGOs together, and we obtained good data, quality data, and quality output, and then together insist the decision makers maybe.
Someday.
Yes,
someday.
Someday.
Yes, but I know that in 60s, 70s, in the first years of 20th century, okay, a family had 6, 7 kids, okay. Now a family has 1 kid, okay. So worldwide, the number of kids in a house are declining, huge decline, okay. But in the same time, the number of world population are increasing. These two issues are contradicting, okay. But the world population is increasing. What happened? We have an increase of population with a huge decline of the number of kids in a family. From 6 to 1. What happened? And we have a world. No, not the worst part, but as an average, the number of kids in a family are reducing largely in the last 100 years. The thing that has changed is we have reduced to 0 the baby mortality. Okay, this is the key point. In fish stocks, we don't have reduced juvenile mortality. Instead, we have increased lots the mortality in the coastal zone. We have degradation in coastal zone. So the number of fish that are enriched the open sea are less and less. And now we are fighting outside, fishers, managers, NGOs, about this low number of fish individuals. But we don't care about the nursery areas where most of the fish needs be growth. Okay, this is the problem. We had a nice slide about growth overfishing and recruitment overfishing with the, okay. We don't respect coastal zone. So how can we say that one person is, we blame this person for not having fish? One other important issue that I would like to comment is, I don't know in Turkey, but also in Greece, of course, but also in the other Western Mediterranean countries, Spain, in Portugal, there is less and less young people enter into the profession. Fishers, young people are reluctant to enter to the profession. And now we have less and less fishers, not because of some side measures, but also because they are not reluctant to work as a fishers. So this is a problem, or this is a good for the fishers management. It's good for the fishers management.
less people,
yes.
Less problem. Yes, in same in Turkey. The younger ones not to work with family as a fisher, so they move to big cities and find another jobs. And now some fisher sports there is no our youngs, but youngs from other countries. Also in academic.
Sorry, that's an endless discussion outside, but because we have another section coming, so that lastly one of them hope that we have to thank again Dr.
I have
give you a lot to do on that.
Yes.
Good morning, everyone. This is a session related with blue economy, development of innovative feed products. And our keynote speaker is Professor Dan C. Vodnar. He's a professor at the Faculty of Food Science and Technology from the University of Agricultural Sciences and Veterinary Medicine of Cluj-Napoca and the president of the Senate within the same university. Since 2016, he has had the privilege of leading an extremely passionate research group in the University of Cluj-Napoca. In 2024, their group was honored as the best research group in Romania in the biotechnology domain during the Romania Research Gala. Professor Vodnar coordinates and has coordinated 6 international research projects and 6 national research projects, focusing on studying the metabolic capacity of probiotic strains and developing solutions to identify substitutes for essential sources of micro and macronutrients. From 2023, he's the coordinator of the Horizon 2020 project Marie Skłodowska-Curie Actions, called FeedActive, which attracted a total funding of €8,610,000. Since 2019, Professor Vodnar has been consistently recognized in the annual ranking of the top 2% of scientists in the field of food science by Elsevier BV and Stanford University. In 2022, he was elected a member of the European Academy. In 2022, he received the Romanian Academy Award Remarkable Scientometric Indicators. For example, H-index 45, 5,169 citations, a brilliant record. Professor Vodnar appears in the top 1% by citations, highly cited researcher with 7 articles. He's editor-in-chief for Bulletin of University of Agricultural Sciences and Veterinary Medicine, Cluj-Napoca, Food Science and Technology with impact factor 0.7. And Professor Vodnar is the author of 5 inventions and 3 patent applications. In 2022, together with his team, he launched HydroMediT, a symbiotic product demonstrating our ability to bring breakthrough innovations to the market. So, Professor Vodnar, after this impressive presentation of you, I welcome you to the stand.
Thank you. So thank you so much for the nice introduction, and thank you for the invitation for joining this wonderful conference. And I have to admit that I'm really glad, keen, happy that you are our partners within this consortium, since I think our consortium put a lot of let's say, happiness within the partners, and the communication between us, it's an excellent asset for all of us. Therefore, I'm highly and deeply I mean, I feel very somehow keen and pleasant for this collaboration. Okay, I'm now going to give some words about the Romanian. Probably most of you knows about Romania, but few of you most probably knows where is Cluj-Napoca, the land we are doing research. It is in Transylvania, in the western part of Romania. And here you have some pictures about the town. Here actually is the rectorate building, and here is our research institute, Institute of Life Sciences, where we are doing our research. So, competing with the Greek gastronomy, the Romanian ones are also somehow very, very tasty, but with a lot of influence from Turkey, from Hungarian. And you will find out many common, I will say, products also in the Romanian cuisine, as in Greece, for instance, is. But what is different, or I like to consider that is different, is the vegetable dip. Our partners, the ones who came to Romania, probably they had the chance to taste it. Sarmale, it is another cabbage wraps are another specialty of the Romanian cuisine. Eggplant salad dip, trip soup, and of course grilled meat. The Romanian values as a nationality or as a population is based on the family. The family is the heart core of the society, and religion is also a very important value of the Romanian. And of course, as you are, all of you, proud of your identity, we as a Romanian, we are also proud about our identity. Nevertheless, the research directions of the group I'm leading, it's more dedicated to the fermentation processes, the fermentation bioconversion of vegetable waste into valuable organic chemicals. And you can see here biovanillin, succinic acid, citric acid, and so on. And optimization of fermentation processes for the cultivation of probiotic bacteria. Another research direction is dedicated to biotics and especially to the microencapsulation of probiotics. Since you all know that the probiotics, they are very sensitive to the pH of the stomach, therefore they need to be protected in order to express their value in terms of activity into the gut. Therefore, we cultivate and produce different probiotic bacteria for food supplements. And we microencapsulate them by using different techniques. And you will see two of them explained in this presentation, very related to our Horizon project. And the specific use of probiotic bacteria in neurological diseases, because nowadays it's very important to focus on the microbiota. And biotics means also antibiotics, but means also probiotics. And the probiotics are nutribiotics, are psychobiotics, and so on. Therefore, we are looking a lot in this direction together with some partners from the medical school of Cluj, but also together with two of my PhD students, because they are running different experiments related to the nutribiotics and neurological diseases. And hopefully we will demonstrate our hypothesis related to this issue. And third, and very important, I will say, the research direction, it's related to the antimicrobial, anti-mutagenic activity of plant extracts and also antioxidant. Nevertheless, since 2020, we decided to set up another direction within the group. It is more related to the molecular gastronomy. And molecular gastronomy actually is the understanding of the behavior of different molecules when you cook a food. Because we eat a lot, but usually we eat faster and we do not know exactly what we eat. And in order to protect, for instance, different nutrients within a food, we shall know about the processes. Anyway, as already Vasilis introduced me actually, we have different products and prototypes released within our university and within our research project, or after having the research project results. And one of these products, it is related to the gut microbiota. And you can see here actually a bottle. And you can imagine that in the bottle cup are stored probiotic bacteria, microencapsulated probiotic bacteria. And in the bottle, we design a beverage made from different pills, because pills are very consistent in prebiotics. The prebiotics are, for instance, different fibers, soluble fibers, oligosaccharides, fructooligosaccharides. Therefore, we decided to make this symbiotic product, and the probiotics are stored in the bottle cup. And when the customer wants to drink this juice, he has to press the bottle cap. The bacteria are released into the bottle. Then you have to shake it and to drink it. And the bacteria we selected in this project was related to the population which suffer by irritable bowel syndrome, together with the University of Pharmacy and Medicine of Cluj-Napoca. And we got good results, not great, because it's a very hard topic, especially for the women over 40 years old. And there are many factors affecting the irritable bowel syndrome. But we got at that time in 2016, we got 52% positive results in with this strain selected. Anyway, this project was also selected by the European Social Innovation Competition as the first 30 ideas across 1,400 ideas from the Europe submitted for that competition. And we were very proud. But this product, it is a prototype right now. We are not selling the product because we produce science more than we produce products. But we have also an ecbiotic. This product, it is a release of the 15, I will say, years of research in the probiotic field. And you can see here in the left, it is a symbiotic product coupling 6 or mixing 6 probiotic strains with a prebiotic. And we sell this product regionally. On the right, you will see a product dedicated for kids, because the kids, they have different gut microbiota, and we selected 2 strains, especially from the kids' microbiota. One was Bifidobacterium breve, and the other one Bifidobacterium infantis. And we microencapsulate these strains, and we include it into a jelly. And this jelly was somehow personalized for my kid at that time, because he was eager to eat a lot of sugar-related product. And I decided to design a more healthy product, let's say. And of course, we patented also these products. Another prototype we have in the lab, it is a sticker for fruits and vegetables which are consumed with pills. I mean, I'm a vegetable lover, and I was all the time when I go to the supermarket, you know that for instance, on the apple, all the time there is a sticker. You have to take out the sticker and then to wash the fruit and to consume. And at that time, I was wondering if there is or if I can design together with the students a sticker to be 100% biodegradable, but also to act as a biodetergent. And the sticker, it's made from biopolymers, sequestrating inside different natural compounds, which have antimicrobial properties. I mean, act as a biodetergent. And when you tap the water and to wash the fruit, for instance, the sticker is going to release or is releasing usually the compounds and act, as I already mentioned, as a biodetergent. On the right, since in the lab I'm coordinating 90% women's and only 10% men's, we decided that for the International Women's Day to create a product for ladies. And we apply different techniques from molecular gastronomy, and we design an edible lipstick, which is made from pectin, for instance, together with some fruits puree, and it was or had a very strong impact into the media. And now, because I was talking about the molecular gastronomy, which has not be confused with food cuisine, here are different products. They are looking, for instance, this egg as a normal egg, but it is made by using different fruits or purees. On the right, you will have Kartoffeln salad together with one student from Germany, which is a very common salad in Germany. We decided to change the shape and the texture of the food, and we get, for instance, another dimension, I will say, of salad, Kartoffeln salad. It is potato salad, actually, and we increase also the cost of the selling this product. I mean, if the Kartoffeln salad, it's a boring, usually food, if you change the shape, if you change the taste a little bit, or to increase the taste, you can sell it with much more money. Anyway, here are different other products. We design a three-dimensional caprese salad. We design a ravioli, vegetable ravioli, and sweet burger, and we use different vitamins in order to create a molecular cocktail, especially for the boys, because they were asking to add vitamin B2 into the cocktail. And when you go into, for instance, into the pub and you have the light, the UV light, the cocktail is going to look or to give this fluorescence. Anyway, going back, this is the presentation of our group. Going back to our very nice project. The project, it is called Development of Functional Fish Feed of Marine and Herbal Origin. And the main goal of this project is the development of innovative aquaculture feed supplements based on natural bioactive compounds in order to boost the immune system and exhibit antioxidant and antimicrobial activity. You will see here that our objectives are related to the development of a new and, more importantly, sustainable source of natural ingredients that can be integrated at an industrial level within the food supply chain. The other objective is related to the customer demands for chemical-free personalized nutrition. And the third objective is the improvement of the productivity of the European aquaculture with environmental, economic, and social acceptance. We are 8 partners. Most of them are here, and I would really like to give a huge thanks to all of you for traveling and for meeting all here. From 3 European countries, it means 314 secondment months. The project is running for 48 months. We have to deliver 14 results, or I mean 40 reports related to our research. The project is structured in 8 work packages and of course has a budget of 1.4 million euros. So as I already mentioned, we are 8 partners from 3 countries. The countries are Italy, Greece, and Romania. And here you will see the name of all the partners. And the project workflow, of course, as I already mentioned, is designed in 8 work packages. The first one is related to the management and organization. The second one, dissemination and communication. The third one is related to the characterization, evaluation, and selection of algae and plants with antioxidant and antimicrobial properties. The work package 4 is related to the protection of these bioactive ingredients with antimicrobial activity and also antioxidant activity in order to be integrated into the feed supplements. The fifth work package is related to the development of innovative feed products by replacing with phytobiotics. The sixth one is related to the evaluation of the effect of selective bioactive ingredients against common bacteria fish pathogens in vitro and of new enriched fish feed on a semi-pilot scale in the simulated condition of cultivation. And here in this building, part of this project it's running downstairs. And the seventh work package is related to the evaluation of the effect of innovative feed products during commercial operation aquaculture conditions on growth, survival, and parasitic load. And of course, the ethical requirements, which are all of them are related to the work package 8. So as I already mentioned, the first research task is related to the selection of edible of bioactive compounds, I will say, from agriculture byproducts, from herbs and from microalgae. Of course, we were having a reference databases and we discussed together as partners, together with all the partners from this consortium, and we decided to stay on the agriculture byproducts as olive leaf trees, citrus peel, and you will see here lemon peel, orange peel, mixture of lemon and orange peel, organic fruits, blueberry leaves, and bog berry. And also we took into account, or we were looking at the herbs, rosemary essential oil, thyme essential oil, and garlic essential oil. And of course, sea lettuce was the microalgae. We found out that it might be very suitable for our intentions, and also chlorella and spirulina. And then we proceed with a collection of plants from different origins, from Greek producers and from Romanian origin, as you can see in the slide. And then we move to the recovery of bioactive ingredients from selected plant and algal species. And we use conventional methods as Stocklet is, but also green methods as ultrasound, microwave, and a combination with the ultrasound and pasteurized liquid extraction, high pressure extraction. And we try to use different mixture of solvents, ethyl acetate, water, and of course, ethanol. And you will see here that after using different materials, different leaves and peels, and the combination of two of them by applying methods I already said, I mean the ones dealing with the green extraction, you can see in the right different colors of the extracts. That means that each method has the potential to extract compounds, which was very normal somehow, because the literature also indicated this. But what was interesting in this step, it was actually the combination of the methods used for the extraction. And you will see here that after having the HPLC results, we were looking on the total flavonoids, and the best results was registered in the lemon water extraction, having a higher amount of flavonoids. Then the total flavonoids, by using Stocklet extraction, you will see that it was very low, considering or reported to the ultrasound extraction. Therefore, the ultrasound extraction for us, it is a great method in order to extract the best flavonoids, which are, I mean, the best amount of flavonoids, which is associated with the antimicrobial and antioxidant activities. Nevertheless, we use the ultrasound-assisted extraction with microwave-assisted extraction, and you will see here that the lemon water followed by the orange, the mixed water, and the orange water was the most suitable combination. Related to the olive leaves, we investigated also the phenolics from the olive leaves, and you will see here that the higher amount of phenolics were released after having the ethyl acetate extraction. And the tyrosol, which is the compound from the phenolic compounds, was in a higher amount in a Soxhlet ethyl acetate extraction. However, the bog bilberry, after a high pressure extraction, recorded different antioxidant activities in terms of total phenolic content, total flavonoid content, and DPPH antioxidant method. Switching to the other agriculture byproducts, I mean herbs, we investigated the rosemary essential oil, and you can see that here, as was expected, the eucalyptol was the major compound found. In the garlic's essential oil, there were different other compounds related to the chemical composition. We investigated also the chemical composition of the essential oil of conhead, on thyme essential oil, and eucalyptus. And for sure, this is a brief, let's say, picture of chemical composition, but very important, I would like to underline, for antimicrobial and antioxidant activity. And here we go with the extraction, and we test the biological activity in terms of antimicrobial activity by using minimum inhibitory concentration. And we use different gram-positive bacteria and gram-negative bacteria. And you will see here in the samples on the left, different extraction methods, combined methods, and also on the right, the results got it for each strain. And at the end, we compare all the results with the gentamicin, which is which was the antimicrobial, a standard antimicrobial and our reference. And as can be seen, the lemon peel ultra assisted extraction in ethanol 80% has a very good antimicrobial activity. I mean, was registered somehow very, very close to the gentamicin for Pseudomonas aeruginosa. And then we moved to another application within the project dealing with the microencapsulation or encapsulation techniques in order to increase the bioavailability of the extracts to maintain the activity through the digestive system, to improve the absorption, to easily handling and incorporation into the fish feed, and to cover, of course, the unpleasant and strong odors of the extracts. And we use the extracts with different matrices, and we did the electrospraying and spray drying methods, or like an encapsulation techniques, followed by the freeze drying. And here you can see some slides where the electrospraying and spray drying are presented as a techniques. And we use then cyclodextrin and starch, and we take into account when we select the matrices. And then we investigate the stability of these nanostructures by using Fourier transform infrared and differential scanning calorimetry. And we are going to do this together with our partners within the next couple of months. And then we will follow the extraction of the solubility of development, micro and nanostructures, and release of these compounds by using HPLC, different spectrophotometrical methods, and gas chromatography. And we moved to the development of innovative feed products by replacing antibiotics with phytobiotics. And we have from our industrial partners, but not only, from the partners dealing with fish growing, two recipes. One it is listed on the left and one on the right, with approximate composition of protein, fat, fiber, and you see here in the slide what I'm trying to offer you or to explain to you. Here there are different necessary activities regarding the fish to be checked or to be followed, and one is related to the oxygen and pH of the water, and also to the checking the vegetation and grass condition of the pools, and trial fishing every few days and checking the condition of the fish. And there are many other necessary activities regarding the quality control, as the incoming raw materials are, and the extruded feed, the quality control of the final product, and of course all the time to look into the EU legislation for the production of feed. One of our partners helps us with life cycle assessment, because it's very important to run this assessment in order to make all the process feasible economically, but also environmental, very important for us. And then this life science cycle assessment at the end will be probably the most important part of the project in order to evaluate how feasible is to be translated or transferred to the whole industry. And our partners developed different questionnaires to collect data from the inventory steps of the LCA, as it is human toxicity and different other issues which are listed here. Going to the evaluation of the effect of selective bioactive ingredients against common bacterial fish pathogens in vitro and of new enriched fish feed on a semi-pilot scale in simulated condition of cultivation. Here we have one of our partners from Italy, Panitica, and they were looking, and you see also the plan for what is going to be run probably in the next months. They design 4 tanks, one filled with sea bream in internal, internal tanks, and outside tanks, and sea bass, of course, following the similar, let's say, matrix. And you will find here different key results obtained and delivered by our partner, with a specific growth rate, with the protein efficiency ratio, with the daily weight gain and feed conversion ratio. And what can be say, it was that Vibrio harvey was the most eminent bacterial strain affecting larger individuals of sea bass. And of course, there are many other, let's say, conclusion until now, but all the conclusion will be delivered, of course, as I already mentioned, in the deliverables. Another facilities, another partner from Romania, Piscicola, they were looking into the carp, and they feed the carp with corn, wheat, and barley. And as can be seen here, the matrices of the results are presented in terms of the proteins, fat, starch, and moisture. And the conclusion of their study until now is that there were no disease-related observation for carp, and differences on the replicate ponds might be the results of variation in the environmental parameters. Evaluation of the effect of the feeds and the feed products during commercial operation aquaculture condition on growth, survival, and parasitic load will be provided for identical tanks. These tanks are suited for the safe and healthy maintenance of the sea bass. They will be weighing 1.5 grams or R. They will be used to test the feed, and one tank will be used as a negative control. There is also the health monitoring of the fishes. We have also a partner from Italy, from the University of Messina, and they are looking for investigated histological examination and also virological one. And they are taking samples for fish and making different experiments. Of course, the dissemination of the project is very important, and our partner is doing a great job, and you will see afterwards some delivery, some actually key indicators which are released within the next moment, I mean the workshop. But we are present on the Facebook, on the LinkedIn, on the Twitter, and if you would like to find out or to find our results or the progress of the project, please follow us on these platforms. Now I think that I have to click if I don't. Okay, so as a delivery or as a dissemination, we already have some articles released, and here is the FeedActive website, and we can share with you our first FeedActive workshop. Now we will have the second one, and how it was. There is a movie here, and I would like to share with you. University of Agricultural Sciences, the Veterinary Medicine of Cluj-Napoca, is proud to be the coordinator of the FeedActive project. FeedActive started in January of 2023, and it's here to bring innovative solutions to the need for fish feed supplements. FeedActive Consortium is comprised of partners of a large spectrum of interdisciplinary knowledge and expertise, 4 research, technology and development organizations, and 4 small and medium-sized enterprises from 3 European countries that exchange skills and knowledge. FeedActive Ambitious is to develop innovative aquaculture feed supplements based on natural bioactive compounds that boost the immune system and exhibit antioxidant and antimicrobial activity. To accomplish this, during FeedActive project lifetime, 314 secondment months will be implemented, training at least 27 researchers in the scientific area of extraction of bioactive compounds, formulation of innovative fish feed, evaluation of their effect in small and large-scale aquaculture, and implementation of overall sustainability assessment. In the future, FeedActive will organize workshops and experience exchange events to bring the project outcomes to a wider audience. FeedActive ultimately aspires to promote young entrepreneurship, self-employment, and innovation in the fish feed value chain
to meet European Union sustainability goals.
FeedActive is here to develop aquaculture feed using natural compounds that boost the immune system of farmed fish.
And last year, within our international conference, it was the researcher night all over the Europe. This is an international, let's say, program. And we were presented also at that time in Cluj, the researcher night. How it works better. So, and I will let you to, and I will just click the slides in order to see some facilities within our university and institute. We were included in our project in terms of infrastructure, of course. And of course, I would like to thanks to all the partners of the FeedActive project, but especially to my team, because without them, this presentation couldn't be possible. Thank you so much.
A really fascinating presentation, Professor Wodner. Thank you very, very much. I liked very much this preparation of food looking like, for example, an egg, and it's not being an egg. Fantastic work, and also a fantastic presentation of what we're doing on FeedActive project. Thank you very, very much.
Thank you so much.
Is there any question from the audience? No? Okay. Thank you very, very much.
We would like to inform you that in 5 minutes, we will be ready to start with our FeedActive workshop. Thank you very much.
I don't know if it was heard, but in 4 to 5 minutes, we will start the workshop on FeedActive. Sorry for the delay to start the workshop. We need to solve some technical problems first. Thank you.